Hello friends, welcome to my course Microbial Biotechnology. We are in module 8 where we are discussing about environmental biotechnology. Today we will be discussing about microbial waste treatment methods. So broadly this lecture is divided into 3 broad sections. In the first section we begin with an introduction, then discuss about the sources of pollutants in nature, generation and accumulation. Then we try to learn how wastes are classified and into what they are classified. Then we take some idea about the projected waste generation in the future, then role of microbes in waste treatment. We will also discuss about natural waste degradation cycles, then microbial metabolism, and then we discuss about biodegradation and necessary recycling systems. Some recent research on microbes in plastic degradation will also be presented. Then we discuss about the significance of biodegradation in waste treatment and bioremediation, and we try to understand the differences between biodegradation and bioremediation. So in section 2 we have microbial waste treatment methods, and in fact there is one more section here, section 3, where we will discuss about the mechanisms of microbial waste treatment. So let us try to understand how big the problem of pollution is. So you can see that everything on this earth today is polluted. We have air pollution, we have land pollution, and we have water pollution. And then you can see the gases coming out of these vehicles in some of the cities, which is a very normal sight, which contributes a lot to air pollution. Then our oceans are also today hugely polluted by the plastic waste that we use. And this waste generation and accumulation is threatening the ecosystems globally. Even remote areas like Antarctica, where there is no habitation, we can find traces of pollutants reaching over there. The Great Pacific Garbage Patch highlights plastic waste buildup in the ocean, and there are many such islands of plastics across the various oceans of the world. Air and water currents disperse waste worldwide, spreading pollutants across borders and affecting ecosystems far from their sources. So, places where there are no industry or no habitation are not actually safe from pollutants because they may be drifting away from other places and sources. So, what are the sources of pollution in nature? We have some natural sources of pollutions which is inevitable and not controllable, which happens due to biological decay, and then forest fires, and then some lightning strikes happen which causes these forest fires sometimes. Then we have the salt spray from the oceans, then we have cosmic dusts, and also many times our desert, when exposed to strong winds and turbulences, will lift the soil, sand will be lifted, and then they will be drifted to far away places. Then there are other things like the radon, fog and mist and ozone. These are all happening due to natural phenomena, and we have very or actually no control on them. But then there are anthropogenic sources of pollution, like which are coming from the industries, power plants, oil refineries, and then the domestic waste, then pharmaceutical waste and agricultural waste, and the automobile exhaust, which we saw you in the earlier slide. So these are man-made pollution. So that's why we call this as anthropogenic, due to the human activities. We can control them to a large extent if we have a proper planning. Now, waste is not one single entity. Some of the waste may be very dangerous. We call them as hazardous waste, and while some may be not that dangerous, we call them non-hazardous waste. For example, the ones which emit radioactivity are very hazardous, and then we also have certain industrial waste which may have a lot of toxic components in it. Either it may be discharged in the water bodies, or it may be discharged through smoke into the air. Then we have the electronic waste, which are happening due to the electronic gadgets when they are out of use and they are damaged, or their life cycle ends, are actually disposed of into the environment. They are now a big cause of concern. Then we have the medical waste, which is again a mixture of many things, which may be the plastic, and then the body tissues, or it may be the soft wastes like the bandages, and then it can be also metallic wastes like the needles of the injection syringe or surgical equipments, etc. So these are all under the hazardous waste because they may cause harm to us in various forms, particularly to our health. Then in the non-hazardous waste we have say for example general municipal waste. Then we may have some other industrial waste which are not hazardous but are coming from the industries. And then we have with these under the municipal waste we may have the organic waste, mostly dead animals or the vegetable wastes coming from the kitchen. And then we have the waste which are generated from the leftover of the packaging materials, that is the packaging waste, and others like materials, glass, plastic, metal, etc. So how much waste is currently produced and how much waste will be produced in the future? So it is very common knowledge that with population increase, the waste is going to increase because people are going to use more commodities, and with the use of increase of commodity, the waste is also going to increase. So this increase in waste is an increasing threat to nature and of course biodiversity. One of the challenges is that where to dump these waste, they require a lot of space. So there is a challenge of really accommodating this waste, which also consumes a lot of land resources, so which could have been used for other alternative use, including parking or converting them into biodiversity parks. So this is some projection in the past 2016 figure when you compare with 2030 which is in the center, and projected waste up to 2050. So you can see that there is an increase in the waste generation in almost all the regions of the globe, starting from Middle East and North Africa to South Asia, which is having one of the largest population of the world, and then similarly East Asia and the Pacific. So our problems with waste is going to increase, not decrease. So therefore we have to think about handling these waste. So one of the ways that we can take care of this waste is through treating them or handling them and processing them and converting these waste maybe to some kind of a wealth, which we call as a waste to wealth approach. So in this approach microbes play a big role. So what are the role of microbes in waste treatment? We can use microbes in the treating waste, and then also these waste treated waste we can use for filling up the land or as landfills. So natural waste degradation cycles are very helpful in this approach. So we have discussed some of these in the earlier lecture. Then biodegradation and bioremediation would be very essential in this entire process, which we will be discussing in the next lecture. And overall microbial metabolism is used to break down complex compounds into simpler compounds, which are complex compounds may again be toxic, and when we break them down they may be becoming non-toxic, and we can probably use these as a raw material for some other industry. So let us start with natural waste degradation cycles or the biogeochemical cycles, which are natural processes that recycle nutrients and waste products in ecosystems essential for maintaining environmental balance. So one of the important cycle is the carbon cycle. The decomposition of organic matter releases carbon, which plants use for photosynthesis. Then we have the nitrogen cycle. Here the decomposers break down organic waste, converting nitrogen into usable forms by the plants. Then we have the phosphorus cycle, where waste breakdown releases a phosphorus essential for DNA and ATP, which plants then absorb as a nutrient. These cycles naturally detoxify ecosystems, recycle nutrients and sustain life by converting waste into usable forms. So, we have covered microbial metabolism in detail in module 3. Let us have a small recap of the basics here. For example, you already know about aerobic respiration, which requires oxygen for energy production. Glucose is broken down to pyruvate, then to carbon dioxide, water and ATP via Krebs cycle and electron transport chain. These are used in activated sludge processes and biotrickling filters, which we will discuss in the later part. Then you have the anaerobic respiration, which does not require oxygen or happens at very low concentration of oxygen. These uses alternative electron acceptors like sulfate, nitrate and fumarate instead of carbon dioxide. Oxygen. Less efficient ATP production happens here compared to aerobic respiration, and it drives processes like methanogenesis in anaerobic digesters. Then we have facultative anaerobes, which can switch between aerobic and anaerobic respiration based on oxygen availability. Some examples are E. coli and Pseudomonas species. These are versatile and adaptable for various waste treatment scenarios. Let us have a peep into nature's own recycling system, which is basically what we call biodegradation, which is a natural process driven by the activity of bacteria and fungi. You can see here in the image one fungi. This serves as a vital means of decay in the environment. It is like nature's built-in recycling system. These microorganisms act as decomposers, breaking down large complex organic molecules into smaller, simpler ones through the action of enzymes. This decomposition process is essential for replenishing ecosystems and ensuring the continuous cycling of nutrients. This process of biodegradation can be divided into 3 stages. The first one is the biodeterioration, involves the alteration of the material's properties at the surface level due to exposure to various abiotic factors like sunlight, temperature and moisture. This weakens the material and makes it more susceptible to further breakdown. Next comes the biofragmentation. This stage is a lytic process where polymers are broken down into smaller units called oligomers and monomers. This can occur aerobically in the presence of oxygen or anaerobically in the absence of oxygen or in low oxygen concentration. The third stage is assimilation. The products of biofragmentation are integrated into the microbial cells. The process provides the microbes with the necessary nutrients for their own growth and survival. Let us now take the example of biodegradation of plastics. Plastics we know are a big challenge today from the pollution point of view, and today the microplastics are contaminating the soil, and that microplastics are also being absorbed by plants. And some of them have been reported to land up in the flowers, and the honeybees have, while sucking the honey from the flowers, actually also suck in the microbes, and there are reports where microplastics are also been found to be now present in honey. So, that is the level of challenge we have today with plastics. So, it is very essential that we think about degrading plastics, which is actually a challenge. So, however, biodegradation of plastics has come as a very interesting idea. Most conventional plastics are highly resistant to degradation. Some microorganisms have evolved the ability to break these tough polymers, which is a big area of research today. So, now let us look into the figure which tells us about the process of biodegradation of plastics. So, these enzymes are attached to the surface and cleave the polymer chains. So, this is organic waste or plastic or inorganic waste maybe. So, then there is excretion of extracellular enzymes by the microbes, and these enzymes actually will go and attach to the surface of these plastics. And in this process it will release carbon dioxide, water and other metabolic process, and surface erosion actually takes place by this enzymatic action and may release water soluble intermediates. So, these intermediates may assimilate into the cell, or there are certain short degradation intermediates which are dissolved into the media. The plastic degrading microbes are able to therefore utilize the plastic as a food source, converting it into smaller molecules that can be further decomposed and incorporated into the environment. So, some of the recent research on microbes in plastic degradation, this is a work published in 2024, where microbial decomposition of biodegradable plastics on the deep sea floor. So, we know that oceans are now full of plastics, and some of them have actually gone down, sunken down onto the ocean floor. So, this team has studied on the decomposition of such kind of deep sea floor plastics. Then we have this study on the biodegradation of polyethylene by the marine fungus P. album. So, here you can see that P. album is actually put into a incubator, and in 9 days they are degrading some of the plastics that are in this incubator. So, basically this P. album was isolated from plastic debris in the North Pacific subtropical gyre. This is one of those plastic islands that I mentioned in the beginning. Then this organism is found to be able to mineralize UV treated polyethylene into carbon dioxide. Over time interval of 9 days, mineralization of the UV treated polyethylene occurs at a rate of 0.44% per day, which is low, but still it is a very good rate considering the challenge of plastic. Despite the high mineralization rate, incorporation of PE derived carbon into fungal biomass is only minor. But then now there are other ideas of genetically engineering these organisms to enhance the biodegradation. Then there are reports of polystyrene degrading bacteria in the gut microbiome of marine benthic polychaetes which support enhanced digestion of the plastic fragments. So, that is a very interesting and happening area of research currently. So, we are trying to solve problems of plastic pollution by first identifying and isolating naturally occurring microbes which can do the job for us through the action of certain enzymes and metabolic pathways. And once we are able to identify those with the means of genetic engineering and as well as genome editing, there is enough scope for enhancing the activity of these strains. So, what is the significance of biodegradation in waste treatment? This helps us in eliminating harmful pollutants, number one, and helps us in recycling the nutrients which are released as a result of this biodegradation, and it actually can reduce the waste volume. So, one of the challenges of pollution is the scarcity of dumping grounds where we can pile up the garbages, and you can see this is a problem in medium to large cities and metropolis. So, if you are able to decrease that volume of waste generated by a city, we will be able to handle it in a better way. Then we can also use these waste for generation of renewable energy. We will have a discussion on these in module number 11. And biodegradation is turning up to be a cost effective method of waste treatment. So, bioremediation uses microorganisms to break down environmental pollutants in soil, water and air. Both natural and introduced microbes are employed, but optimal conditions including nutrients, pH buffers and minerals are needed to be worked out. The process of bioremediation relies on oxidation reduction reactions with oxygen, nitrate, sulfate etc. as the electron acceptor and organic substrates as donors. So, in the next lecture we will be discussing about bioremediation in detail. Some of the steps involved in bioremediation techniques are as follows. In step 1, we identify and study native microorganisms with high pollutant degrading potential. In the second step, we go for cultivating microorganisms sustainably in the laboratory conditions. And in step 3, we assess organisms, microorganisms pollutant degrading abilities, their efficiencies through bench scale experiments. And in step 4, we monitor bioremediation progress using chemical analysis and toxicity testing of the products produced by these process. Let us now try to understand the differences between biodegradation and bioremediation. So, biodegradation is a naturally occurring phenomena. Bioremediation is developed by humans, so it is man-made or it is an engineered process. The target of bioremediation is wide range of organic matter. In bioremediation, since it is man-made, the target is specific pollutants or contaminants. Biodegradation is a passive approach, do not require human intervention. If we allow some waste to stay in a space, natural forces will take care of that. The process may be slow, but over time the biodegradation will start decomposing that material, and which may range from day to weeks to months to years to decades. Then we have these bioremediation process. Here the approach is active, we require human interventions. Majority times we will be developing reactors where we will treat the waste, or we also construct dump sites, and we have various gadgets and infrastructure for degrading waste by this approach. So, that is why it is an active approach. So, some of the examples of biodegradation, which is a natural process, is composting and decaying leaves. The leaves falls from the tree, we do not touch it, it stays there for long and it gets decayed on its own over time. Some of the examples of bioremediation, which is a man-made and which is active process involving human intervention, are cleaning up oil spills and then treating wastewater which may be generated by the industry or by the city waste or from the city waste. Let us now move on to section 2, where we will be discussing about microbial waste treatment methods like suspended growth systems. Under that we will be studying about activated sludge method, then sequencing batch reactors or SBRs. Then we will be discussing our attached growth systems, where we deal with biotrickling filters, rotating biological contractors. Then we have moving bed biofilm reactors. Then we go for membrane based systems, where we discuss about membrane bioreactors, membrane anaerobic reactors, and also microbial fuel cells. Then we have another section 3. Let us move to microbial waste treatment methods. So, we can divide them broadly into 3 types: suspended growth systems, attached growth systems, and membrane based systems. So, under suspended growth systems we have the activated sludge method, and then we can have sequencing batch reactors. In attached growth systems we have biotrickling filters, rotating biological contactors, and then we have moving bed biofilm reactors. In membrane based systems we have membrane bioreactors, then membrane anaerobic reactors, and also microbial fuel cells. So, let us look into the suspended growth systems, which rely on microorganisms free floating in the wastewater to break down organic matter and other pollutants. So, the advantages of suspended growth systems are that they are highly efficient, and it helps us in rapid degradation of the organic matter. Adaptability is another advantage, suitable for various waste streams with different compositions and strengths. Comparatively, they are very simple to operate, and they require less complex infrastructure compared to other systems. Then the flexibility is one of the advantages, due to which it is easy to modify and scale up or scale down systems, and they are very cost effective. So, we have lower operational cost compared to some other technologies. However, they also suffer from certain disadvantages. like they consume high energy. It requires continuous aeration for aerobic systems. Then there is excess sludge production, therefore requires disposal or further treatment of the excess biomass. Then it may be sensitive to shock loads. Sudden changes in waste characteristics can disrupt the microbial activity and therefore actually decrease the efficiency. These systems are susceptible to bulking, formation of filamentous bacteria, can hinder settling and reduce the treatment efficiency. What are the different types of suspended growth systems? Number one is the activated sludge method. This is the most widely used system. It employs aeration and mixing to maintain a high concentration of active microbes. Then we have the sequencing batch reactors or SBRs. These SBRs are advanced wastewater treatment systems that operate in a batch mode, allowing for a sequential process of filling, reacting, settling and decanting within a single reactor. How activated sludge works? So, here you can see the schematic of an activated sludge system, and here is a inlet through which we will send in the waste, and this is the aeration tank where we send in compressed air. You can see here from this point, and then the waste will be degraded in this particular tank, and then the degraded products will go to the clarifier, and here the sludge will be deposited, and this is the outlet through which the overflowing liquid will go out, and this sludge is extracted through this opening. Now, this sludge may contain material which is not fully degraded or treated. So, we may actually recirculate it and put it back into the aeration tank, and further we expose it to air for aerobic degradation, and these process can continue for a couple of times until and unless we get a very good degradation. So, now let us see into the operations. In the aeration tank, we introduce air to support the microbial growth. Microorganisms consume and break down the organic pollutants in the aeration tank, and then there is the sedimentation where settling of the activated sludge takes place. The sludge is recirculated, a portion of settled sludge is recirculated for sustained microbial activity. Then we have a secondary clarifier where separation of treated water from the settled sludge takes place. Let us look into the working of a sequencing batch reactor. So, here we have the influent, mixture and air. So, we fill it up which is static, mixed or aerated, and then we have a in the step 2 the reaction where the mixing or aeration takes place, and we allow the reaction to happen for some time interval, after which the sludge will settle down. And then we decant the top water and separate out the sludge, and then this waste sludge is allowed to be idled, and then we again may fill up with additional influent material, and this cycle can actually go on. So, in the first step, which is a filling step, inflow wastewater into the reactor. Then in the next process, there is a biological treatment and neutral removal, and settling or sedimentation of the solids take place, and by decanting we remove the treated water, and the idling period is between draw and fill the time interval. And if we compare both the two methods of activated sludge versus SBR, we can see that two processes are different. So, from the operational point of view, activated sludge is a continuous operation, okay, and then but here it is not continuous. This is basically batch wise cycle. From the vessel point of view, we have separate aeration and settling tanks. In the case of SBRs, single reactor for all the phases. We have moderate flexibility in the case of activated sludge. In the case of SBRs, we have highly flexible cycles or phases. Then we have less control on activated sludge process, but in SBR more precise control over each phase. From the points of view of energy consumption, activated sludge require very high energy because of the continuous aeration, but SBR the consumption of energy is lower because aeration takes place only during the reaction phase. Then we have the effluent quality both high, and in the case of SBR there is potential for higher quality. Then excess sludge production is very high in the case of activated sludge, but it is comparatively lower in the case of SBRs. From the point of view of cost, activated sludge require lower capital cost, but higher operational cost because of the continuous need for energy. SBR, we have a higher capital cost, but we have lower operational cost. From the application points of view, activated sludge is most common municipal and industrial settings for water treatment. SBRs are suitable for smaller communities, fluctuating influent flows and high effluent quality requirements. Let us now discuss about the attached growth systems. These systems are powerful and versatile category of biological wastewater treatment methods. However, unlike suspended growth systems where the microorganisms are freely suspended in the wastewater, attached growth systems utilize a fixed surface for microbial colonization. This leads to the formation of biofilms, communities of microorganisms encased in a self-produced extracellular matrix. So, what are the advantages of using attached growth systems? They are also highly efficient. Biofilms can achieve high removal rates for organic matter, nutrients and other pollutants. They consume very low energy, requires less aeration compared to suspended growth systems. The small footprint requires less space than some suspended growth systems due to high biomass density. They are resilient to shock loads. They can tolerate fluctuations in wastewater flow and pollutant concentrations because of this biofilm nature. They are simple to operate and maintain, requires minimal maintenance in fact. Again, like any other system, they also have certain disadvantages. For example, they have a slower startup. Biofilm development takes time until and unless the microorganisms are arranged and encased in a extracellular matrix to form a biofilm. They won't be able to show their metabolic activity as a group. So, this leads to longer startup times than some suspended growth systems where degradation may start quite early. There is a clogging potential. Biofilm growth can potentially clog the media, requiring periodic cleanup or replacement. Then they are sensitive to toxic compounds. High concentration of toxic chemicals can harm the biofilm. Limited control over microbial population. The composition of the biofilm is determined by environmental conditions and may not be easily controlled. How does these attached growth system works? Number one is there is a wastewater distribution. Then secondly, biofilm formation and growth takes place. Then there are organic matter degradation by the diversity of microorganisms in the biofilm happens. Then we have the oxygen supply and aeration. These are typically open systems that allow natural air circulation. In some cases, forced aeration systems supplement natural air circulation and optimize treatment performance. The treated water collection happens after this, and we have to maintain the biofilm. As the biofilm grows thicker, a portion of it naturally sloughs off the media and exits the filter with the treated waste. As a result, trickling filters require secondary sedimentation to remove the sloughed biofilm measured as suspended solids. Additionally, periodic cleaning or replacement of the media may be necessary to prevent excessive clogging and ensure optimal performance. So, what are the different types of attached growth systems? We have trickling filters, we have rotating biological contactors, and then we have moving bed biofilm reactors. In trickling filters, wastewater trickles over a bed of media, allowing biofilm formation and degradation. So, here is the schematic of trickling filter. So, you have these sprinkler on the top, okay, which is sprinkling the media wastewater. Then there is the filter layer here, as you can see. Then there is a feed pipe over here. Then this is the filter support, which separates the collection chamber from the media chamber into which we have these filter. Then natural air is allowed in, as discussed earlier, and this is the outlet through which the treated wastewater will exit. So, this is a simple arrangement in a way. Now, we have the rotating biological contactors. So, this system is a static system, it does not move. In the RBCs, we have rotating discs with biofilm growth. These are partially submerged in the wastewater for treatment. So, some of the system components are an rotating biological system consists of a circular tank containing a series of rotating discs mounted on a central shaft. So, this is the side view of this system, and this is the front view of this system. So, you have these media discs or panels stacked next to one another, and from the front view you can see this is the central shaft, and this is the central shaft, and there is a motor which drives this shaft. So, here you can see the media support around the disc, and these discs part is in the air, and around 35 to 40% is submerged. So, the disc is rotating alternately between air and the liquid which is getting treated. So, this is partial submergence of the disc allowing the formation of biofilms on their surfaces. The rotation speed of the discs can be controlled to meet specific treatment needs. So, that has to be optimized. So, since the disc is moving here, so this is rotating biological contactor. Now, we have the moving bed biofilm reactor. This is a system where freely moving media with biofilm growth are fluidized in the wastewater. Biofilms grow on media within a fixed bed or packed column reactor. So, this is the schematic of a moving bed biofilm reactor. Basically, you have a reactor 1 and a reactor 2. So, in the reactor 1 we introduce the raw effluent for pre-treatment, and then we have the air blowers for aeration from below. Then in reactor number 2 we have the sedimentation, and there also we have this air distribution grid. So, here we have the specially developed media loaded, and the treated effluent is taken out from this outlet. So, this is one of the special kind of one design which we call as the moving bed biofilm reactor. Now, what are the difference between trickling filter versus RBC and MBBR? So, in from the design point of view, we have already shown you trickling filter is stationary bed or media. A rotating biological contactor have rotating discs with biofilm. In a moving bed biofilm reactor, we have freely moving media with biofilm. Some of the advantages of TFE is simple low energy resilient, cost effective. RBCs have high surface area, compact, good nitrification, and it requires again low energy. In MBBR, we have the capacity for high biomass density. It is compact, efficient, and adaptable. However, each of them has certain disadvantages. Trickling filter, slow startup, order limited, and then nutrient removal is a challenge. There is a problem of clogging. RBC also there is mechanical complexity, disc breakage may happen, and there may be bulking of the filaments. Then in the MBBR we have very high cost of operations. There is a media loss, and we need to mix the things regularly. And then from the suitability point of view, trickling filters are suitable for small medium communities, industrial settings, and bioremediation applications. And rotating biological contractors are useful for municipal, industrial, high strength wastewater treatment. Similarly, MBBR also useful for municipal, industrial, and while upgrading plants, and for high strength wastewater treatment. Let us now have a discussion on membrane based systems. So, these combine biological treatment with membrane filtration for high quality effluent and increased biomass retention. Some of the advantages are, these are high quality effluent, meets stringent discharge standards, and can be reused or discharged to sensitive environments. It has reduced environmental impact, minimizes sludge production and land requirements compared to conventional methods. Improved public health, reduces the risk of waterborne diseases by effectively removing pathogens. Resource recovery enables the recovery of valuable resources such as biogas and water from waste streams. However, there are certain disadvantages like high initial capital cost. Membranes and associated equipments can be very expensive. Membrane fouling may occur. The membranes get clogged by organic matter. Then high energy consumption, requires energy for both aeration and membrane filtration. There is sensitivity to toxic compounds. High concentrations of toxic chemicals can damage the membranes. And then there is operational complexity, requires more sophisticated operation and maintenance. What are the different types of membrane based systems? So, we have membrane bioreactors which combine activated sludge treatment with membrane filtration for high quality effluent and reduced footprint. Then we have membrane anaerobic reactors. They utilize anaerobic digestion with membrane separation to treat organic waste and produce biogas. So, this is the schematic of a membrane bioreactor. So, this is a submerged membrane bioreactor here, and this is the side steam membrane bioreactor. Then we have a rotating disc membrane filter over here, and you can see here the biological membrane is inside in this case, and while here it is outside, and then you can see the inlet position from here. But in this side steam membrane bioreactor, the sludge is recycled and the sludge passes through this biological membrane. In the case of a rotating disc membrane filter, we have rotating disc covered with biological membrane, and there is a shaft, and of course, there will be a motor which will help us in rotating it. And then the wastewater enters from one side, and this disc keep on rotating, and the treated waste is taken out through the outlet. So, this rotating membrane filter was developed by Reed et al., which consisted of a pressurized membrane with hollow rotating shaft and hollow membrane covered disc stacked along with it. This design led to the reduction of membrane fouling and cost. How does a membrane bioreactor works? Wastewater enters the biological reactor, as you can see in both the cases from the top, and here microorganisms in the activated sludge degrade organic matter and pollutants. Mixed liquor is pumped to the membrane filtration unit. This contains the treated wastewater and the activated sludge. Pressure is applied on the mixed liquor. This forces the water and dissolved molecules through the membrane, leaving behind a concentrated stream of solids and microorganisms. Then the treated water is collected, the permeate or treated water is collected and can be discharged or reused. Concentrated solids are recycled or disposed. The retented or concentrated stream containing the biomass is recycled back to the biological reactor to maintain the desired concentration of microorganisms. Excess biomass is then removed. A portion of the concentrated solids is periodically removed to prevent excessive accumulation and maintain treatment efficiency. Membrane anaerobic reactor and its working mechanism. So, here you can see the schematics of a membrane anaerobic reactor. So, you have a membrane here, and then this is the anaerobic bioreactor, and these pump will force this material to be treated through this membrane. The permeate will be going out, while the retented will be recycled back into this system. So, organic waste like food scraps and agricultural residue enters this anaerobic digester. So, this is the feed which may be any of these waste. Then the microbes will decompose the waste, producing biogas, methane and carbon dioxide, and also the digester. Then digester is sent to membrane filtration unit, separating it into permeate, that is the treated effluent free of solids and microbes for discharge or reuse. The retented, the concentrated solids and microbes are recycled back to the digester to maintain microbial concentration. Biogas is collected for use in electricity generation, heating, or as a fuel. Let us now have a discussion on microbial fuel cell briefly. We will discuss about these in detail in later lectures. Microbial fuel cells are recognized for their capacity to generate electricity through microbial metabolism, utilizing various degradable wastes, which is quite an interesting technology. So, here you can see the schematics of a microbial fuel cell with the cathode at one end and anode at another end, and then electricity is produced due to the reactions over here, and then this is actually basically having microbes loaded into it. The concept of microbial fuel cells date back up to 1912, when Potter demonstrated electricity production from Saccharomyces and Escherichia coli, and later in 1931 Cohen claimed nearly 35 volt electricity production. Let us now discuss an interesting system which is produced as a result of membrane filtration process combined with biological processes and electrochemical processes. We call this system as a bioelectrochemical membrane reactor system. So, there is a growing interest in combining these microbial fuel cells and membrane bioreactors for water treatment and electricity generation. Bioelectrochemical membrane reactors or efficiently generate electricity and remove organic pollutants with recent design advancements, enhancing performance and reducing costs. Let us now go to section 3, where we will be discussing the mechanisms of microbial waste treatment, including biomineralization, biosorption, biotransformation, bioaccumulation and bioleaching. So, the various mechanisms of microbial waste treatment falls into biomineralization, biosorption, biotransformation, bioaccumulation and bioleaching. But then we have various types of biomineralization, which are microbial induced carbonate precipitation, microbial induced phosphate precipitation, microbial induced sulfide precipitation. Then we have heavy metal biosorption, and then we have heavy metal biotransformation and heavy metal bioaccumulation. Then we have autotropic bioleaching and heterotropic leaching. So, microbial induced carbonate precipitation, here urea hydrolysis by urease bacteria produces carbonate, which reacts with heavy metal ions in the soil, forming precipitates that immobilize heavy metals. So, here you can see the cellular metabolism producing urease, and then these helps us in hydrolyzing urea, and then forming the precipitate and immobilize the heavy metals. So, these are the heavy metals, as you can see over here. Now, in microbial induced phosphate precipitation, phosphate mineralization is caused by bacteria which produces phytase or phosphatase, and that hydrolyze organic phosphorus into phosphate. Phosphate biominerals then form with heavy metal ions in the soil, fixing the heavy metals. So, you can see the production of phytase and phosphatase, which helps us in MIPP biomineralization. In microbial induced sulfite precipitation, sulfate reducing bacteria utilize sulfate as an electron acceptor. So, this is an electron acceptor, and the organic substrates are the electron donors to oxidize the organic compounds, producing sulfide, and it increases the alkalinity. The generated sulfides react with metal ions here, forming metal sulfides that precipitate and immobilize the heavy metal. Let us now discuss about bioabsorption. Bacteria, algae and fungi act as biosorbents, with bacteria excelling due to their surface to volume ratio and binding sites like carboxyl, phosphate, hydroxyl groups. Several studies have found that dead bacteria often outperform living ones in bioabsorption, as shown by Bacillus sphaericus, which had 13 to 20% higher chromium ion absorption when dead. This may be due to resilience to toxicity, no nutrient requirement, storage without degradation, and cost effectiveness. So, heavy metal bioabsorption by bacterial biomass. The figure shows the mechanism of bioabsorption. So, there is a precipitation that involves chemical interaction between the cell surface and the metal surface in step A. Then in step B, there is complexation of heavy metals that occur by interaction of the metal ions with the surface active group of bacterial biomass. Then in step C, we have exchange of bivalent metal ions which occur with the counter ions present on the bacterial cell in the ion exchange mechanism. And then we have diffusion in step D. This is a simple process of bioabsorption without the involvement of any rate limiting step. So, this enters the cell inside. Then there is also surface adsorption, which is a rapid and reversible process in which metal cations bind to the bacterial surface and ions via non-specific attraction forces. And then we have intracellular accumulation of heavy metals that take place by the transport of metal ions across the cell membrane. Let us now discuss about the biotransformation process, which involves the active metabolic conversion of contaminants into less toxic forms by microorganisms. Through metabolic pathways, bacteria and fungi break down organic pollutants like hydrocarbons, pesticides and industrial chemicals. This process can result in complete degradation or mineralization or conversion to less harmful substances, facilitating easier removal. So, here this figure shows the biotransformation of heavy metals during which they are converted to less toxic form. So, these heavy metal ions may be accumulated inside, which is called intracellular bioaccumulation, or they may be immobilized onto the cell by covalent bonding or ionic bonding, or there may be complex formation and chelation, and that can happen after internalization, or that may lead to the internalization, and there is also a cation exchange happening over here, and then many reactions are shown over here where inorganic arsenic is converted to organic arsenic, chromium 4 is converted to chromium 3, mercury ion is converted to elemental mercury, and these are considered as the less toxic forms. Now, another method is the microbial bioaccumulation, which is a metabolically active process where heavy metal ions are transported into the cell via transporter proteins involving mechanisms like endocytosis, ion channels, and carrier mediated transport, and we have discussed these in module 3 earlier. So, this is the schematics for the process of bioaccumulation, where these heavy metals are internalized, and this is the extracellular polymeric substance as shown in the blue color. This process allows microbes to store heavy metals, thus remediating polluted environments. However, extremely high concentrations of contaminants can overwhelm microbial defense systems, leading to cell damage or death. Let us now have a brief discussion about biolysing. Biolysing mainly is performed by acidophilic microbes, involves the oxidation of ferrous to ferric and sulfur to sulfuric acid in low pH environments. This produces ferric ions and protons, dissolving metal sulfides and oxides from ores. Microbes are used to extract and recover heavy metals or other soluble contaminants from soil by converting into soluble substances for removal. The process can be direct bioleaching or indirect bioleaching, and both have different kinds of mechanisms. So, in the direct bioleaching, microorganisms directly attach to the metal surface, as you can see here in this picture. The bacterial cell interacts with the mineral, enzymatically breaking down the sulfide bonds. Electrons are transferred from the metal sulfide to the bacterial cell during the oxidation process. This leads to the release of soluble metal ions, example ferrous, cupras into the solution. This process is driven by enzymatic activity on the mineral surface in brief. In the indirect mechanism, microbes generate ferric ions and other oxidizing agents in the surrounding solution. These oxidants, example ferric ion, reacts chemically with the metal sulfides, as you can see here, causing them to oxidize and release metal ions. The role of bacteria here is primarily to regenerate ferric ions from ferrous via metabolic processes. These ensures a continuous supply of oxidizing agents for metal solubilization. So, with this we come to end of today's lecture. Thank you for your patient hearing.